Hi all, this is Miss Solomon, and I'm going to talk to you about blood pattern analysis. What is blood, right? Blood is a type of connective tissue that we have in our bodies that helps us transport a bunch of different things around our bodies. You might think of red blood cells when you think of blood. I'm going to show you a picture on the next slide, but this is not the largest component of blood. We can talk about red blood cells. Red blood cells are the largest amount of cells in our blood. They help us carry oxygen to and from the lung, or from the lungs to the body, and then go back to the lungs to gain more oxygen. They have hemoglobin, which helps carry that oxygen through binding. Okay. It also has a bunch of enzymes, but red blood cells don't have any DNA in them, so they cannot be used for DNA analysis. Also in our blood, there's white blood cells that are part of the immune system. The liquid of the blood, the things that are not cells, but it's liquid, right? So it needs to have a liquid component. That is going to be plasma. When you separate the cells out of plasma, it's actually yellowy. And there's a bunch of water in there, plus a bunch of dissolved things. So what's dissolved in there? Electrolytes, nutrients from your digestive system, vitamins, hormones moving around your body, carbon dioxide that's been dissolved, so it's in a special form called bicarbonate, a bunch of different stuff. There's antibodies from your immune system. This is the majority of blood, is actually the liquid, not the cells. Additionally, there's platelets. Those are also dissolved in the blood plasma, and these are protein fragments that help with clotting. Okay, those are all of the parts of the blood. So if we were to take a sample of blood and you centrifuge it, which means you spin it really, really fast, the dense parts separate from the less dense parts, and the dense parts move to the bottom. And so what you end up having is something that looks like this, where you have 55% of your blood is plasma, the liquid part. A little teeny portion is white blood cells and platelets, and then 45% is red blood cells. So a significant portion is red blood cells, but the majority of your blood is not blood cells. Okay, so what are the blood types? You might have heard of blood types. You may or may not know your blood type. We have groups that we are categorizing blood types based on a type of antigen present on the outside of the cells. Okay, this is based on your genes that you inherit from your biological parents. The blood types that exist are A, B, AB, and O. And then for each of those, you can either be positive or negative. And so you can do blood typing to determine what your blood type is. And again, you get these from your parents. So if your parents know their blood type, your biological parents, through your genes, this is determined based on genetics. So a lot of people ask, like, what's the most common blood type, or is this rare or common? So this is approximate worldwide distributions of blood type. You'll notice that A positive is really common at 31%, and also O positive is incredibly common at just over 40%. Okay, those are the two most common blood types. Positives in general tend to be more common than the negatives of that same type. So AB positive is more common than AB negative. O positive is more common than O negative. Negative is a recessive genetic pattern. So it makes sense that in many cases, positive is more common than negative, though that's not always the case with recessive and dominant genetics. And then certain ones are incredibly uncommon: O negative, B negative, AB negative. So if you were to find blood like that at a crime scene, that would be something very interesting, because you could really narrow down a list of suspects based on blood type. Okay, so this says blood type cannot be used to prove guilt, but it can be used to establish innocence, right? So just because you found A blood at the crime scene and a suspect has A blood, that does not necessarily mean that they committed that crime, because a bunch of people can have A blood, right? However, if you found A blood at the crime scene and a suspect has type B blood, that could be used to establish their innocence in that crime. So this is a type of class evidence, right? You can use it to narrow down suspects, but it cannot be used to prove guilt. Okay? So when we do find blood at a crime scene, here's another one. Here, when we do find a stain of blood, or what we think is blood, we need to test it. So here are some tests for blood. We will do all three of these tests in class. So there's the hydrogen peroxide test for blood, where the blood will bubble and create gas. There's the Kastle-Meyer test for blood, where it will turn pink in the presence of blood. And there's a luminol test, which I'll talk about in a second. The thing to know about these, especially the first two, is that they are presumptive tests, right? Which means that they can only establish that it is either totally not blood or it could be blood, right? So if those tests test negative, then it is definitely not blood. If they test positive, then it's likely to be blood, but it's not a guarantee. So after you do a presumptive test, then you might do a confirmatory test. Those tend to be more expensive or more in depth. And so you don't want to do those confirmatory tests, the more expensive tests for every single thing, right? We would only do it after it's proven positive on a presumptive test. We will do the hydrogen peroxide and Kastle-Meyer tests in a lab. And we might do a luminol test at our crime scene. Some blood stains are latent, right? They are not easily present. Maybe someone went through and cleaned this. That is what that luminol test is for, okay? Even after someone goes through and tries to clean with some like heavy duty cleaning chemicals, some evidence still remains. Tiny particles will cling to surfaces for years. And someone might never even know they are there, okay? So the chemical luminol, that is one, I'm very much simplifying this, but when it gains an oxygen, it will have the opportunity to glow via a process called chemiluminescence, right? It's glowing due to chemistry. This sort of process is used in glow sticks, right? The chemicals interacting allow it to glow for a certain period of time. Okay? I'm not going to get too much into the in-depth of how that works, all right? But the thing to know is that it specifically, in this case, is interacting with hemoglobin, which is that compound in red blood cells that carries oxygen. And so because hemoglobin is pretty stable, this test can detect blood stains that are even several years old. However, it only lasts for a short period of time after you use it. And so this would be a test that we would do either if we're not sure or after we've done other presumptive blood tests.